This is Biology 100, Chapter 3, Chemistry of Life. So I want you to remember as we're going through this PowerPoint, any concepts or terms that are underlined and blue should be written down and become a part of your growing study guide. Let's begin. The objectives for our chapter this morning are to understand the basic structure of an atom, to compare and contrast the types of bonds used to form molecules, discover how hydrogen bonds produce the unique properties of water, explain the difference between hydrophilic and hydrophilic molecules, and between acidic and basic molecules, and understand the 4 types of macromolecules, what they are made of, and how they function in living organisms. Let's begin with matter, anything that has mass and occupies space. So mass is a scalar quantity. It only gives us information about the magnitude or the amount of stuff in an object. It is not always about the size. A brick on the scale would have, if it was made out of lead or made out of felt, it measures the same amount of mass. So mass is measured in grams, and a gram is about the weight of a paperclip. Or it can be measured in kilograms, and 1,000 grams would be a textbook. Or it can be measured in tons, which would be about the size of a car. So don't mistake mass for weight. A force that depends on the mass of an object and acceleration of gravity. On the moon, you are the same mass, but a lighter weight than if you're on Earth. An element. An element is a pure substance that has distinctive physical and chemical properties and that cannot be broken down by other substances or substances by ordinary chemical methods. Some examples of elements that we know are hydrogen, oxygen, carbon, sulfur, phosphorus, etc. So we'll look at the periodic table of elements and we can see hydrogen, oxygen, helium, carbon, and all of those many familiar elements. So the periodic table here arranges elements by the number of protons that they contain. The protons are what's found inside the nucleus of an element, and we'll talk more about that in a moment. So hopefully you're able to see H is hydrogen, O for oxygen, Fe for iron, Na for sodium, C for carbon. Many of the letter designations make sense, like O for oxygen and He for helium, and others are confusing because they're based on the Latin phrase. So for iron, Fe stands for ferrous instead of for iron. Okay, I'd like to take a moment to refer back to our biological hierarchy diagram, which can be found on page 14 in chapter 1 of your textbook. If you've not done so already, please make sure that you've put a Post-it note on this page, and that because we'll be referring back to it for the remainder of the semester. Please notice that the first level on the hierarchy are atoms. Atoms are the building blocks of all matter, living and non-living. We begin our exploration of biology understanding the smallest particles that we can perceive. So an atom, like I just mentioned, is the smallest unit of an element that retains its distinctive properties, the building blocks of matter. There are 2 parts to an atom. We have the nucleus, which is inside the atom, and around that we have the electron clouds. And those shells get bigger and bigger and allow for more electrons to grow as we move out. The outermost shell of an atom is called a valence shell. And that valence shell wants to be full. If it's full, it's at its most stable. So atoms can gain or lose electrons from other atoms to try and fill its shell. And that's where we start getting the combination of atoms together to make molecules. Now, some examples are noble gases have a full outer shell: helium, argon, neon, krypton, radon. Their outer valence shell is completely filled. All other elements have partially filled valence shells. In that innermost shell, you can have 2 electrons. In the second shell out, you can have 8 electrons. In the third, you can have 8. In the fourth, 18, and so on and so on. Let's take a look at an atom. So here you can see in the center, the nucleus. Inside this nucleus, we have neutrons as well as protons. The neutrons are gray and the protons have the plus mark on them. You'll find the electrons here in the shells. So here in the first shell, you're able to see 2 electrons. You go to the next shell and you see 4 electrons. Okay? So the nucleus is positively charged and it's bound by those protons and neutrons pulling together. A proton has a positive charge. The neutrons are just like what we're saying, they're uncharged, they're neutral nuclear particles. And the electrons have that negative charge, and that's what's surrounding and keeping that energy going around that innermost proton. Your atomic number, this is very important, it's unique to each element. Your atomic number is going to be the number of protons in an atom's nucleus. So, for example, if we're looking here, we'd see that the hydrogen has 1 proton because the atomic number is 1. Here we can see that carbon has 6 protons because the atomic number is 6. Nitrogen, 7 protons, and oxygen, 8 protons. The next number that we have that we can see in that square is the atomic mass. This is approximately how much the atom weighs. This is the combined number of protons and neutrons in an atom's nucleus to get the number. They're not always the same number of protons and neutrons. So if we look at this atomic number here, with hydrogen, we can see that we have 1 proton, 0 neutrons, okay? Within our carbon, we can see that we have 6 protons because we have the atomic number here, and there are 12 combined neutrons and protons. So we're able to see that there are 6 neutrons because we can take 6 away from 12 and get the 6 neutrons. Again, here we have the same amount in nitrogen, where there are 14 total protons and neutrons. If we take that atomic number away, which is the same number as our protons, then we'll know that we have 7 neutrons. An isotope forms an atom that has the same number of protons, but a different number of neutrons. So that's where we have what's called an isotope. Again, we have protons with a different number of neutrons. Okay, let's continue on. So here we have a question. Nitrogen, let's see, how many protons, electrons, and neutrons does the hydrogen atom shown here have? Okay, so we just talked about that. Let's look at it. We have one proton, we have one electron, and we have 0 neutrons. The atomic number is the number of protons, and the atomic mass number is the number of protons plus neutrons, and both are 1. How about for carbon? Again, we just went through that. So there's 6 protons, 6 neutrons. So we have 6 is our atomic number, and our atomic mass is 12. So carbon-14 is an interesting idea. So carbon-14 is something that is found in some different kind of specific living organisms. And this is an isotope of carbon. So the isotope in an atom is where we have the different number of protons and neutrons. So if we were looking at carbon-14, we would see that we have an atomic number of 6. So therefore we know that there are 6 protons. If it has 7 protons, it wouldn't be a carbon atom, it would be a nitrogen atom. But here, when we're looking at carbon-14, there's 6 protons and then 8 neutrons. So it has an atomic mass of 14, making that an isotope of carbon. Now let's look at nitrogen-11 as an isotope of nitrogen. So we see that nitrogen alone would have 7 protons with an atomic mass of 14, so it would have 7 neutrons. Now, since it's an isotope and it's called nitrogen-11, we're going to know that the atomic mass number is in fact 11. So how many neutrons are going to be in this isotope? We have 7 protons, 4 neutrons. Okay? Molecules. This is a molecule is the next step on our biological hierarchy. And here we're looking at when 2 atoms or more bond together. Elements always want to fill their outer shell, and they're that outer valence shell. So they're attracted to other elements in preparation, trying to fill that shell. Okay, so different kinds of bonds. A covalent bond is formed when 2 atoms share an electron. This is going to be your strongest bond. A molecule is an atom linked by covalent bonds. So here we can see we have 2 hydrogen atoms. That first valence shell is able to take on 2 electrons. So by forming together, they're filling each other's shell by having those 2 electrons. An organic molecule. This is very important that we understand of our organic molecule. Any molecule that contains at least 1 carbon-hydrogen bond. So organic compounds are very important in our daily life, and they range from simple to extremely complex. Organic molecules make up a big part of our own bodies. They're in the food that we eat, and they're in the clothes that we wear. Organic compounds are also used to make products that we use: medicine, plastic, washing powders, dyes, and so many other things. There are millions of organic compounds that are found in nature, but we can also make them synthetically. The most important organic compounds that we will discuss this semester are proteins, lipids, carbohydrates, and nucleic acids. Carbon atoms. Carbon is found in all living things. Carbon atoms move constantly through living organisms, the ocean, the atmosphere, and the Earth's crust, and this is all known as the carbon cycle. Carbon has a unique property, has unique properties which influence how it behaves and how it bonds with other atoms. So for one, carbon has 4 valence electrons. This means that each carbon atom can form with a maximum of 4 bonds. So if we can remember, we want that second shell wants to have 8 electrons in it. When it only the carbon atom only has 4 electrons in its second shell, therefore it can bond with 4 different elements. Because of the number of bonds that carbon can form with other atoms, organic compounds can become very complex. Number 2, carbon can form bonds with other carbon atoms to form single, double, triple covalent bonds. Carbon can also form bonds with other atoms like hydrogen, oxygen, nitrogen. Carbon can form, number 4, carbon can form in a straight chain. It can be branched, or it can be in cyclic molecules. This is all really important because how those molecules are formed is how they're going to be useful in what that molecule needs it to do. So if we look at these compounds, we can see carbon dioxide, that's in a straight chain. Propane is going to be in a branched formation, and methane is in that cyclical molecule. So it's all, how are these different molecules formed together to have a reaction? And also, how stable are they going to be? A chemical reaction. Also extremely important that you know this for the remainder of the semester. It will become particularly important when we put it into application into some of our upcoming labs. So, chemical reaction is the process of breaking existing chemical bonds and creating new ones. So what we're putting in are the reactants, the beginning compounds that undergo a change during that chemical reaction. So reactants are going into the chemical reaction. Products are the compounds that are made as a result of that chemical reaction. So reactions go in, products come out. So here we can see in CO2, we have carbon and we have oxygen are going into, they're going to have a chemical reaction going into that chemical reaction, and what we get on the other side is carbon dioxide on the other side. Let's look at another example of a chemical reaction. Here we have photosynthesis. So plants and algae undergo photosynthesis to create their own food. We're really going to talk about this in a short while. So start thinking about these different elements. What we just saw, that carbon-oxygen combination of carbon dioxide mixed with 6 water, H2O molecules, and sunlight, at the end of it, that's what's going into, those are our reactants going into our chemical reaction. On the other side, our products are going to be glucose and oxygen. So let's say that our tree is that arrow in the middle. That tree is collecting carbon dioxide, water, and sunlight. That tree is going to process that water, and on the other side, what we're getting from the tree is glucose, which we use for energy, and oxygen, which we use to breathe. The product is glucose and oxygen. Our reactants, what's going into the tree, carbon dioxide, water, sunlight, products, glucose, oxygen. Okay, let's move forward. Our early Earth's atmosphere. So in your chapter, you read about Harold Urey, who proposed that Earth's early atmosphere resembled that of other planets in our solar system. So Jupiter, Saturn, Uranus. Scientists have often wondered about how life began. In order for life to evolve, organic molecules such as proteins, lipids, nucleic acids, and carbohydrates, remember those macromolecules, needed to be present. The atmosphere of early Earth was much different than what we see today. It was 78% nitrogen, 21% oxygen. Those were the two most abundant gases in, those are the most two abundant gases in our Earth today. The oxygen buildup is a result of the photosynthesis. Methane, ammonia, hydrogen, gases, and water vapor are produced by volcanic eruptions. Early Earth had much more volcanic activity than what we see today. If you refer to your book, figure 3.2, we see Miller's original spark experiment. In 1952, Stanley Miller added methane, ammonia, hydrogen gases, and water to a sterilized apparatus. He heated the water until it became water vapor, which traveled up and over and mixed with the other gases. Miller added an electrical spark to provide energy for the chemical reactions. So let's break that down. Our reactants, what's going into this? We have water, we have the methane, the ammonia, hydrogen. That's what's going into our chemical reaction. Those are our reactants. The chemical spark is what's causing the chemical reaction. Now let's see what the products are. What did Urey find on the other side? So early Earth had a lot of lightning. That's what he was replicating with that electrical spark. When the water vapor cooled, it condensed and collected, and it left behind a deep red and turbid broth at the bottom of the tubes. Miller analyzed these liquids and discovered 5 types of amino acids. Amino acids are the building blocks of proteins. Remember our macromolecules? Proteins, that's a big one. 3 of the amino acids were among the 20 that make up all proteins in living organisms. Miller had shown that the building blocks for proteins could have been produced under the conditions present in early Earth. Proteins are essential to all living organisms. So our product coming out of this experiment is going to be the amino acids. Reactants going in, water and the gases. The chemical reaction happens with the electrical spark, and then our product are these amino acids that are collected. 50 years after Miller's experiment, there was a box of vials that was discovered in his old lab. The vials contained dried samples from Miller's experiments. These, combined with other detailed lab notes that Miller kept, provided valuable information. Technology has improved in 50 years since Miller's experiment, and when the samples were reanalyzed, scientists found 14 amino acids in the original samples and 22 amino acids in the samples where Miller added steam to the apparatus. So, before the experiment was run, the apparatus was sterilized, then carefully sealed. Why is this an important thing to do you suppose? Well, having a spotlessly clean apparatus was important to ensure that any amino acids that were produced by the experimental conditions was made from the experiment, not from contamination. So what he's really trying to make sure is that the only reactants going into this experiment are added through the experiment, not from any pre-existing stuff living within that, that glass apparatus. Next, we have polar molecules. The polarity of water molecules is the cause of water's unique properties. So polar molecules, when the electrons are unequally shared between atoms, partial electrical charges result within this molecule. Water atoms are held together by covalent bonds, which unequally share electrons. Water is made up of 2 hydrogen and 1 oxygen atom held together with those covalent bonds. Oxygen doesn't share the electrons equally, and the electrons spend more time around the oxygen atom. This gives the oxygen end of the water molecule a slightly negative charge, and the hydrogen end of the molecule a slightly positive charge. Oxygen holds the electrons more closely than the hydrogens. So let me say that again. Oxygen holds on to those electrons more closely than the hydrogens do. The water molecule itself has no overall charge because the positive end is canceled out by the negative end. But it creates a polar molecule and gives water some of those unique characteristics. And water molecules are attracted to other water molecules. So, something that you could try at home is to take a glass of water. Take a razor blade or a paper clip and put that horizontally with the flat end carefully onto the water so that you're not breaking the surface of the water. What you'll see is that paper clip or that razor blade will float. Now take that same paper clip or razor and turn it on its side so that that sharp end or the skinny end, that will break through the atoms and it will sink. So let's talk a little bit about what we're observing there. We're seeing how the water is able to create cohesion. Cohesion is the hydrogen bonding and the polarity of the water molecules. When those bonds are being held, they're going to stay together. However, those bonds can be easily separated. So why is this important that water has this characteristic? Well, it's really important for some living organisms like water striders and spiders that walk across that water. So water is referred to as the universal solvent because there's so many things that can dissolve into water. Water can break apart or dissolve other molecules such as salt because water is a polar molecule. Salt is made up of sodium, Na, and chloride, Cl, ions that are bonded together. An ion is an atom that has lost or gained an electron and therefore has an overall charge. If an atom loses an electron, then it has a positive charge. If it gains an electron, it has a negative charge. Salt is held together by ionic bonds. The opposite charges or ions are what is bonding them together. The positive end of the salt molecule is attracted, which is the sodium, is attracted to the negative end of the water molecule. The negative end of the salt molecule, the chloride, is attracted to the positive end of the water molecule. And the water is able to dissolve or break apart the salt molecule. So let's look at these bonds again. We have the three types of chemical bonds that you're going to be responsible for understanding. Covalent bonds are formed when two atoms share the same electron. Ionic bonds are formed when the charges of positive and negative ions hold the atoms together. A hydrogen bond is a weak electrical attraction between partially positive hydrogen atoms and partially negative end of the polar molecule. Hydrogen bonds hold different water molecules together. In order for water to be able to change temperature, these hydrogen bonds have to be able to form or to break. Looking at solutions. This is another set of terms that are really going to want to become a part of your language here in biology, and particularly when we're working in our applied lab space. So solution is going to be a combination of a solute and a solvent. If you think about it as a chemical reaction, your solution is your reactant. Going into that reactant, you have your solute and your solvent. Your solute is going to be the particles that dissolve within a substance. So if we look back at the last slide, your solute is your salt. Your solvent is the liquid into which the solute dissolves. So the solvent is the water. So you have your solute, what dissolves into water. You have your solvent, which is what the liquid that's taking on that dissolving. And those are your products. They go through a chemical reaction and you have a solution, which is going to be the combination of your solute and solvent. So again, this is a very important relationship to understand. Okay, now how does that relate to hydrophilic and hydrophobic? Let's look at that. Hydrophilic is water-loving, a substance that dissolves in water. So it's going to be the solute that loves to dissolve within the solvent. Hydrophobic is water-fearing. Phobic is fear. It's a substance that does not dissolve in water. So I want you to think about this oil and vinegar picture that we have here. The vinegar could be water as well. So the molecules of oil, of a lipid, are hydrophobic. They do not want to dissolve inside of that water. Instead, they're going to clump together. There are many substances that do want to dissolve in water that are going to be hydrophilic. You have salt, sugar, alcohols. Fats do not want to dissolve in water. They just don't mix. The oil will always stay on top of the water because oil is less dense than water. So another at-home experiment that you can try is to take a glass, or you could take a plastic bottle, put water into that, then take oil and put that right on top of the water. Either close the cap or put your hand over the top of the glass and shake it up. You're going to see the two substances mixing, but what you won't see is them staying that way. You're going to see that oil rising, forming together, and then rising to sit on top of the water. Another experiment that you can do is to look at hydrophilic, is to take your glass of water and put a couple drops of food coloring into that water. You're going to see that water dissolve out into the food coloring, will go out into the water, and eventually that water is going to dissolve and take in that coloring. Okay, so what will happen to the molecules of olive oil if you shake the bottle and leave it alone for an hour? What about the molecules of vinegar? So like we just talked about, if you shake this together, you're going to have the beads of oil float right back to the top of the bottle. And then what we'll see is that the vinegar will sink to the bottom, or that water will sink to the bottom. Shaking the bottle will distribute the vinegar molecules and the oil molecules, but they will remain in that solution. So what would happen if you add another fat to the bottle, such as bacon grease, and shook it up? Bacon grease is a fat, and like the olive oil, it's going to form small beads of fat that are going to float to the top of the bottle. So they will even separate from the olive oil. Now, given how sugar behaves when it's mixed into coffee or tea, would you predict that it is hydrophobic or hydrophilic? If you believe that it's going to be hydrophilic, you are correct, because the sugar is dissolving inside of the coffee or the tea. So let's break that down for a moment and look at the different pieces of this chemical reaction. We have sugar, which is going to be your solute. You have the coffee, which is going to be your solvent. And after that chemical reaction, you're going to have your sweetened coffee or your sweetened tea. That's going to be your solution. Okay, so let's talk a little bit more about these unique characteristics of water, because we can find it in 3 different physical states depending on the temperature. So water is unique because as a solid, it's less dense than it is in a liquid state. So that's when we find ice floating on top of water. This is an important characteristic because if ice was more dense than liquid water, lakes and ponds would freeze solid in the winter. Fish, larvae, amphibians, and other organisms would freeze. Ice floats on top of water and acts like an insulating blanket. The water below is actually warmer than the freezing. So to freeze, we have to be at 0 Celsius or 32 degrees Fahrenheit. And those stable hydrogen bonds become crystallized. All of those atoms slow down. And that's where we have that solid form. So ice is hydrogen bonds, and the water molecules are really pulled far apart. So the ice is expanding when it freezes. Everything slows down and goes apart. They're slowing down because of their distance being apart. Now you get above 0 degrees Celsius or 32 degrees Fahrenheit, and what happens? We have those hydrogen and oxygen elements are starting to get closer and closer together. And the closer they get together, the more they're going to start moving around. And so then what we have is it becomes a liquid. And those hydrogen bonds are constantly breaking and forming and going to different bonds. And that's what's allowing for there to be that flow of water, is it's really those hydrogen bonds attaching to different oxygen elements. So then we have water in its gas state. So that's steam. And what we have there is those water molecules are now very close to one another, and the hydrogen bonds are just jumping from one to the other, one to the other. So in order for water to go from liquid to a gas, the hydrogen bonds have to be broken, and it takes energy for hydrogen bonds to break. And that energy is coming in the form of heat. And this is why water doesn't change temperature very quickly. Water slowly warms up and then slowly decreases down. Why is this important? I want you to think about how water plays a very important role throughout our seasons. So we want water to slowly warm up in the winter and then slowly decrease in temperature during the winter time, so that the animals, us included, are able to adapt to those changing cycles. Okay, so I talked a little bit about this cohesion. So the hydrogen bonds that hold water molecules together produce cohesion. This allows the water strider that we see here to walk across the water but not actually break the surface tension. It's the cohesion that produces the round beads of water on a leaf. And when you read the amount of water in a graduated cylinder or in a measuring cup, like you will for lab, you'll always read the middle of where that cohesion is. So because the edges of water will curl up on the sides of the container. So again, when you're measuring water in a graduated cylinder, you're going to the middle of where that arc is. Okay, Miller's hot water injected into the spark discharge experiment enabled a wider variety of chemical reactions to occur. We have evaporation, and this is the transition from liquid to a gaseous state, which requires heat. Then we have the opposite reaction of condensation. So this requires a cooling, and the molecules reform into hydrogen bonds, and then they return back to their liquid state. In this next picture, you're able to see all three states of water in this one picture. So you can see ice as it freezes in the background. You can see the lake in its liquid background. And then you can see the steam coming up off of that lake as it's a a warmed space. So water can be found in all three states of matter throughout the earth, and this is important because life needs water. We as humans are about 70% water, and we can survive longer without food than we can without water. So, light from volcanoes. Miller's inclusion of hydrogen sulfide also enabled a variety of chemical reactions to occur, likely similar to early Earth's chemical reactions. 6 of the 22 amino acids in Miller's test tube contained sulfur. Sulfur is that rotten egg smell when you go past something that's decomposing, such as a marsh. But also you find a lot of sulfur is let off in volcanoes. So an erupting volcano releases all of the gases that Miller included in his mixture, and the volcanic ash contains iron and other metals. So steam is produced when the magma, or the superheated rock, comes in contact with groundwater, and the lightning bolts lance through the clouds of gas and ash. Or is life from outer space? So here's another idea. Scott Sanford of NASA's Research Center produced amino acids from gases and conditions found in interstellar space off of this asteroid, which crashed into the desert in Sudan, which carried amino acids thought to have been produced in space. So amino acids are found in left and right-handed versions, and they mirror each other. Only left-handed versions are produced by living things. And the asteroid had both left and right forms. The right-handed ones produced only in space. Okay, let's change gears for a moment. And now we're going to start talking about the acidity or the baseness of these different elements and compounds. So pH is when we're measuring how well can a substance take on more hydrogen ions. That's what we're measuring when we look at a pH scale. So acids and bases are on chemical opposites of each other, and they can neutralize each other with how many ions they can take or they can take in or that they can give away. So acids are able to give hydrogen ions to their surroundings. Bases accept hydrogen ions from their surroundings. Let's look at the scale here. So there, 14 is our most basic. That means that we are able to take on hydrogen ions. Acidic means at 0, means that we're giving away hydrogen ions. And right there at 7, that's our neutral. So we want to think about that as being water. And then we're going to start moving either which way, depending on the acidity. So here's our scale, 0 to 14, 7 is neutral. And what we'll find is acid is going to be the citric juices, rain, coffee is going to be more acidic. Base or alkaline are things like baking soda, cleaners, and acids. The pH scale is a logarithmic scale, so every one number is a change factor of 10. A solution with a pH of 5 is 10 times more acidic than a solution with a pH of 6. A solution with a pH of 4 is 100 times more acidic than a pH of 6. So When you go from 4 to 5, that's 10 times. And then when you go from that 5 to 6, 10 more. So you have 10 times 10, and that's where you get the 100 times more acidic. Acids and bases will react with each other and produce salt. The lower the number, the more acidic and dangerous the solution is. The higher the number, the more alkaline or basic, the more dangerous it is. Very strong impure acids and very strong impure bases are both dangerous and can burn your skin. So let's ask a couple questions here. Which has a higher concentration of free hydrogen ions? Vinegar with a pH of 2.8 or milk with a pH of 6.5? Which has a higher concentration of free ions? So, a high concentration of free ions corresponds to a low pH. Thus, vinegar has a high concentration of free hydrogen ions than milk has. Next, what happens to the concentration of free hydrogen ions in your stomach when you drink a glass of milk? The concentration of those free hydrogen ions decreases, so the pH increases. Number 3, black coffee has a pH of 5. Does adding coffee to water with a pH of 7 increase or decrease the concentration of free hydrogen ions in the liquid? The concentration of free hydrogen ions increases. So let's go to life's first steps, and we're going to go back to what we talked about in the very beginning, macromolecules, or there also can be called biomolecules, organic molecules. They are all meaning the same thing. So biomolecules, organic molecules, macromolecules. Those terms can all be interused when talking about these larger life-building molecules. So the 4 that we're going to discuss, again, are proteins, carbohydrates, nucleic acids, and lipids. These are all critical for living cells. All are built on the framework of covalently bonded carbon atoms. Remember, carbon is found in all living things because it so easily can form with other elements. Polymers are long strands of a repeating unit of small molecules called monomers. So if we're looking at a long chain, if we had a beaded necklace, each of those beads on our necklace would be called the monomers. The polymers are the whole chain of beads put together. Proteins, carbohydrates, and nucleic acids are all polymers. They're all made up of individual monomers. Lipids, however, are not polymers because their structure is not composed of a chain of monomers. We will get to what lipids are formed from in a moment. So it's very important to understand that proteins, carbohydrates, and nucleic acids share the same structure of monomers creating polymers or long strands. Lipids do not. So here we have what I was talking about with that necklace is that you have monomers here, and then you have your polymers and those long chains. So mono means one unit, poly means many. So Many units strung together make your polymers. Okay. So our biomolecules are critical for life. Let's talk about each one of these 4 macromolecules individually. So if we start here with proteins, there's different combinations of 20 amino acid monomers, and they all allow for making different types of protein. So when we look at amino acid, an amino acid is one of those beads, one of those monomers, and they can be strung together in different ways to make different kinds of proteins that have different functions and jobs. So for example, when we talk about COVID-19 and the proteins that we find in the spikes, they are looking for another type of protein in a receptor. And unless those proteins can find that receptor, you won't have a connection. But the proteins in those spikes on that virus are connected to and looking for the proteins that are found in the receptors. So all of that chemical reaction is happening because of the proteins being specifically chained. What we'll also notice is that proteins are folded into a 3D shape. So they don't stay in that chain. That chain begins to coil around itself and becomes a specific and unique shape that also is a part of its functionality. Let's see. Enzymes are also made up of amino acids and are proteins. Enzymes speed up chemical reactions. So remember our chemical reactions. We have our reactants in, our products out. An enzyme is that arrow in the middle. It's going into whatever is happening in that chemical reaction and making it happen faster. So that's the function of an enzyme, which is a protein, is to make chemical reactions happen faster. So hormone and receptor proteins like insulin and its receptor allow our cells to take up sugar and use it for energy. Membrane transport. So in and out of a cell, and we're going to call that outer cell our membrane, there's different proteins that are looking for different substances to bring in and out of our cell. And without those protein receptors, which is just like I was talking about with the virus, we are looking for those cell protein receptors. We can also use these molecules for storage. Proteins also give us the color of our hair, our skin, and our eyes. So protein has a very wide variety of what it's really giving us. But they vary in size and in shape and in function. So we can use protein for structure, we can use protein for enzymes, for storage, for membrane transport, for antibodies, and toxins. Proteins are extremely important. Next, let's move on to carbohydrates. So this is where we get our energy sources. And carbohydrates are used for energy by cells. Simple sugars or monosaccharides can be bonded together to form disaccharides. Polysaccharides are complex carbohydrates and are formed when many sugars are bonded together. Glucose is a simple sugar. Sucrose is a disaccharide. So we have our glucose. If you remember back, we were talking about chemical reactions. when we're going through the process of photosynthesis, one of the products is the simple sugar glucose, which can be broken down easily by cells. And then there's all kinds of other examples of sugars that can be found that are carbohydrates. So you have lactose, which is found in milk, fructose, but they all end in ose, which is an easy way to tell if a molecule is a carbohydrate. So simple sugars are a cell's direct fuel to make ATP. And ATP is the molecular energy source that's essential for all cellular work. Again, when we talk more specifically about photosynthesis, we will talk more about what this ATP is and how important it is for our bodies and our cells to have the carbohydrate input. Some other carbohydrates such as glycogen, which is in animals, and a starch in plants, are also used for energy storage. So there's a difference there between energy that's being used immediately and energy that's being stored. So carbohydrates can provide structural support to cells through cellulose. And cellulose is a fiber that you're going to find in the cells of plants. So when you're looking through that microscope and you're seeing the cellular walls, which are going to be in that onion root, or that rectangular cellular walls, that's made out of cellulose. And that's what's allowing the form of the plants to have strength enough to be strong and without having a skeleton. That's what allows flowers to grow in the stems to hold up, is the cellulose that's there found in the cell walls, which is made out of carbohydrates. You can also, insects, spiders, crustaceans, they all have exoskeletons, and that is going to be made from carbohydrates. So it's the next most versatile biomolecule. So you have proteins and then carbohydrates. They vary in size and shape and function. So we want to think about carbohydrates being used for short-term energy storage, a cell's direct fuel to make ATP, and for structural support. Okay, third most important are nucleic acids. These are different combinations of deoxyribonucleotide monomers, also known as DNA, and combinations of ribonucleotide monomers, which is RNA. These are really important when we start talking about genetics and how these nucleic acids are forming together. So don't get too stuck on them for right now, but you need to understand that when we start talking about DNA and passing on DNA, that how these nucleic acids line up is going to be extremely important for how genetic material is passed on. So there's two types that form, like I just said, the DNA and the RNA. And they can both vary in size, shape, and function. DNA provides living organisms with long-term, stable genetic information storage in a form that can be easily copied and passed on to future generations. Our genes are DNA. RNA comes in many forms and plays many roles, but its most important job is in providing readable genetic language that enables genes to be expressed as proteins. And then finally, we come to the macromolecule lipid. So different structures of various types of lipid, triglycerides, phospholipids, steroids. they all allow to perform different functions. So when we talk about fats, people tend to have the reaction of fats are not good for you and we should not, and we want to reduce fats. So fat is going to be your long-term energy storage. So lipids are not made of these repeating monomer chains, if you recall me saying at the beginning. Lipids are organic molecules because they have carbon bonds to hydrogen molecules. Some lipids, like the triglycerides, which have 3 fatty acid molecules, are linked to another glycerol molecule. That's what is your long-term fatty storage. Whoops, I went forward one. Sorry about that, guys. And you have phospholipids, which are composed of phosphate groups. So these are hydrophilic structures. You have the hydrophilic, which is your polar end, and you have a hydrophobic, which is the tail end. So that's really what they look like. If you look here at the bottom of this photo, you're going to see that red ball at the top. That's considered the head, and that's your hydrophilic. It likes the water part of the lipid. And then you have the inside purple tail, which is the hydrophobic end. And that is going to be creating a membrane-like structure where you have, and because the purple end, that hydrophilic end, doesn't want to be near the water, so they're going to attach towards one another, whereas the red orange hydrophilic is going to be searching for those water molecules. So when we're looking at their structure, we can see that they have very different structures, and because they're going to float on top of water, they have that energy source that our water-filled cells can use, put away for later, and use when we need it. So when we were running around and hunting and gathering, and we needed those long-term energy sources, they were there for the long, cold winters. So now we might not need them quite as much, and they have now become a problem. So we have to balance out where we are evolutionary in this biology of how we look at lipids. Okay, so the 4 biomolecules that are critical for life. We have proteins, carbohydrates, nucleic acids, and lipids. You do not need to know the chemical formula or structure of biomolecules. You need to know that monomers for 3 of the biomolecules that are polymers and what their functions are, and how they support the 6 characteristics of living things. Those 6 characteristics of all living organisms can be found back in chapter 1. This is another figure that I would put a Post-it note on and really make sure that you understand. And then find the connections between how these biomolecules support those 6 characteristics of living things. Matter makes up everything in our universe. So going back, looping back around to our very first slide of what is matter. And it's the stuff that we are made of. And it's how it's all cycling together to keep life functioning. So what's really amazing, if we take a moment to think about it, is everything in the universe is composed of matter. It's made of atoms. And it consists of all different types of particles. Some we know, some we don't. And that's the most that we can know, is that there's common elements that compose the world around us, that we live in, within our human bodies, within our Earth's atmosphere, and within the universe. So let's move on to clicker questions. Please recall that clicker questions are all, this is all fair game for future tests. Okay, ordinary table salt is composed of one negative and one positive ion. Which of the following types of bonds hold these ions together? C, ionic. The chemical interactions that cause atoms to associate with each other are known as chemical bonds. When an atom loses or gains electrons, it becomes respectively positively or negatively charged ions. Ions of opposite charge are held together by ionic bonds. Covalent bonds are formed by the sharing of electrons between atoms. The molecule contains at least two atoms that are held together by covalent bonds. Hydrogen bonds are weak associations between two molecules, such that a partially positive hydrogen atom within one molecule is attracted to the partially negative region of another molecule. The partial electrical charges result from an unequal sharing of electrons between atoms, giving rise to polar molecules. The polarity of individual water molecules and the hydrogen bonding across water molecules explain nearly all the special properties of water. Number 2, how do the two hydrogen and oxygen molecules in a water molecule interact to create a polar molecule? The answer is A. The oxygen nucleus pulls the negative electrons away from the hydrogen nuclei, creating a separation of charge in the entire molecule. So positive protons remain in the nucleus. Negative electrons can be drawn closer to the nuclei in some atoms, leaving a positive and a negative end to the molecule. Number 3. 3 of the following groups of molecules share an important structural characteristic that the 4th group does not share. Which one is different? Lipids. So all 4 of these are biomolecules, but only proteins, nucleic acids, and carbohydrates are polymer structures that are long strands of those repeating monomer units. Number 4, what makes lipids structurally different from proteins, carbohydrates, and nucleic acids? So I'm asking the same question in a different way. They are not polymers. Again, all 4 of those are biomolecules, but it's only proteins, nucleic acids, and carbohydrates that are made up of polymers. Those lipids have the hydrophilic and hydrophobic membrane. And that is all for our slideshow today for our presentation. I hope you have a better understanding of how chemistry is so important for how our understanding of biology. This is as far as we will go into the chemistry unit of understanding biology, but it's very important that you understand the importance of these small smaller pieces in that larger biological hierarchy. Thank you so much for your time, and I hope you have a wonderful day. Heard that. Oh, I'm sorry.